Today, we're diving into something that sounds like pure science fiction, the ability to literally cut and paste the code of life. We're going to break down the amazing tools and techniques that make modern biotechnology work. So, let's get into it.
But let's start with a question you might not expect. Have you ever baked a loaf of bread? Or maybe made your own yogurt? Or, hey, even just enjoyed a nice glass of wine?
If you said yes to any of those, then guess what? You've already dabbled in biotechnology. Seriously, for thousands of years, humans have been using living things like yeast and bacteria to make stuff we want. And that, at its heart, is all biotechnology is.
But that old school way of doing things, it's a bit of a gamble. As you can see on the left, traditional breeding is slow, it's not very precise, and you often get a bunch of traits you don't want along for the ride. But modern genetic engineering on the right, whoa, that's a complete game changer. It lets us be surgical, targeting and picking out only the specific genes we're interested in.
So how in the world did we make that leap from a game of chance to precision engineering? Well, the story really kicks into high gear with a major breakthrough back in 1972, all thanks to a couple of pioneering scientists. And this didn't just happen out of the blue. You had two brilliant minds working on parallel tracks. In 1969, Herbert Boyer figured out that certain enzymes could act like tiny scissors, cutting DNA in a really specific way that left these sticky ends. At the same time, Stanley Cohen was perfecting a way to move little rings of DNA, called plasmids, in and out of bacteria. Then, in 1972, they put their ideas together. Boyer's scissors met Cohen's delivery system, and bam, they created the world's first piece of artificial recombinant DNA. And here's the really mind-blowing insight. They realized they could turn simple bacteria into microscopic factories. Just like the quote says, they could pop in a new piece of DNA, a new blueprint, and the bacteria would just start cranking out whatever protein that blueprint coded for. This one idea, right here, is what the entire biotech industry is built on. Okay, so if you want to perform this kind of genetic wizardry yourself, you're going to need a toolkit. Think of it like a biological arts and crafts project. You'll need some scissors, a delivery truck, and a little bit of glue. First up, your molecular scissors. The technical term is restriction enzymes. These things are incredible. They are proteins that will scan miles of DNA code looking for one very, very specific sequence. And when they find it, snip. They make a perfect cut. Now, check this out. The truly clever part is that they don't just cut straight through. The enzyme makes this staggered cut, which creates these little single-stranded overhangs we call sticky ends. And they're called sticky for a reason. They naturally want to pair up with a matching edge, which, as you'll see, is absolutely crucial. All right, you've got your gene cut out. Now what? How do you get it to where it needs to go? For that, you need your delivery vehicle, and it's called a plasmid. These are just small, circular pieces of DNA that bacteria have hanging around, and they are perfect for carrying our new gene into a host cell. And here's what one of those plasmids looks like on a map. You'll see it has a few key parts. There's an origin of replication, which is basically the start button telling the cell to make copies, and it also has these marker genes, in this case for antibiotic resistance. This is a super clever trick that lets scientists easily see which bacteria actually accepted the new plasmid. Okay, last piece of the toolkit. Your new gene's sticky ends have found the plasmid sticky ends, and they've paired up. But you need to make that connection permanent. That's where our molecular glue comes in. It's an enzyme called DNA ligase, and its whole job is to seal up the gaps in the DNA backbone, making it one solid recombinant molecule. So, we've got our scissors, our delivery vehicle, and our glue. Let's put it all together now and watch the whole cut and paste operation from start to finish. The process really just boils down to 3 steps. Step 1, cut. The trick here is you have to use the exact same restriction enzyme on both your gene and your plasmid. That way, you know the sticky ends will match. Step 2, ligate. You mix them all together and DNA ligase does its thing, gluing the gene into the plasmid. And finally, step 3, transfer. You get this new, custom-built DNA into a host cell, and your factory is officially open for business. So let's just walk through that one more time. You see here, we cut the DNA we want. We cut the plasmid open, we join them together, and then we put that new molecule into a host cell. And the really powerful part is what happens next. Every single time that little bacterium divides, it makes a perfect copy of our custom plasmid. That process, making millions of identical copies, is called gene cloning. Now that's awesome, But what if you don't have enough of your gene to even start with? What if you only have a tiny microscopic sample from a crime scene or an ancient fossil? How do you make enough copies before you even start cutting and pasting? Well, for that, you need a biological photocopier.
Let's say this is all you have to work with. Just one single molecule of DNA. And in just a couple of hours, you can turn it into this, well over 1 billion identical copies.
How is that even possible? It's thanks to an incredible invention called the polymerase chain reaction, or PCR. It is, for all intents and purposes, a biological copy machine that lets you take one tiny piece of DNA and amplify it into a massive sample, all inside a test tube.
And it works using a simple 3-step cycle that just repeats over and over again. First, you heat everything up to split the DNA into 2 separate strands. Then you cool it down so these little markers called primers can attach to exactly the spot you want to copy. And third, a special enzyme swoops in and builds a new copy. And with every cycle, you literally double the amount of DNA. And the recipe for this is shockingly simple. You just need 4 basic things: your original DNA template, the primers to mark the start and end points, that special copying enzyme, and a big supply of the A, T, C, and G DNA building blocks. That's it. That's all you need.
When you put these tools together, recombinant DNA and PCR, it's not an exaggeration to say they have changed the world. They're the basis for so much, from developing new vaccines and medicines to creating crops that can survive drought to solving crimes. We truly have the tools to rewrite the code of life itself.
And all of this leads to one final, really big question. We've gone from using life more or less by accident to designing it with incredible intention. So now that we have the power to edit the blueprint of almost any living thing, what do we build next?